Hello, everyone, and welcome to Dana-Farber's Department of Data Science, Frontiers in Biostatistics webinar. My name is Erica Fike, and I'm the program manager for the department. If you have a question for the speaker or the organizers, please use the Q&A tab or raise your hand to ask your question live. Our faculty organizers will manage the Q&A session during and after the talk. With that, I will turn it over to Nabia Teo.
Thanks, Erica. So today I have the pleasure of welcoming Dr. Rebecca Hubbard to our Frontiers in Biostatistics seminar series. Dr. Hubbard is a professor of biostatistics at the University of Pennsylvania and the deputy director of the Division of Biostatistics and a member of the Abraham Cancer Center. She received her master's and degree in epidemiology from the University of Edinburgh, in applied statistics from Oxford University, before doing her PhD at the University of Washington in 2007. She then worked at the National Alzheimer's Coordinating Center in the University of Washington and at now currently Kaiser Permanente Washington Research Institute before joining the University of Pennsylvania. She chaired the biometric section of the American Statistical Association in 2018. Dr. Hubbard's research focuses on the development and application of statistical methodology for studies using observational data from community medical practice. This work encompasses the evaluation of screening and diagnostic test performance, methods for comparative effectiveness studies, and health services research. A wealth of data on healthcare utilization, performance, and outcomes are available from electronic health medical records. However, the complex observation scheme giving rise to these data necessitates careful study design and analysis to ensure that the inference is valid. Dr. Hubbard's methodological research emphasizes the development of statistical tools for such inference and has been applied to studies of cancer screening, aging, dementia, pharmacoepidemiology, women's health, and behavioral studies. And today we're going to learn more about this from her in her talk titled Considerations of Extracting Real-World Evidence from Real-World Data. Please join me in welcoming Dr. Hubbard.
Thank you, Nabia, and thank you, Erica, and thank you to everyone who is joining us this afternoon. So it's my pleasure to talk with you today about a topic that I'm quite passionate about, which is how we obtain valid and reliable evidence from real-world data. So throughout this talk, I'm going to use the term real-world data in a very broad sense to refer to data that's collected as a secondary byproduct of transactions that are digitally mediated. So I will principally be focusing on electronic health record data, and a lot of times I'll refer to EHR data interchangeably with real-world data, but I think it's important to keep in mind that EHR data is really the tip of the iceberg in terms of real-world data. And I think in coming years, we're going to see more and more use of additional data sources like claims, social media data, wearable device data. I've got my Apple Watch on right now. All of these things are going to be of more and more potential use for generating evidence about health and health outcomes. So in my talk today, I'm going to tell you about some of the things I think about in terms of how we get valid evidence out of these messy data sources that were absolutely not collected for research purposes. And I was thinking about the fact that this talk was originally scheduled for me to deliver it back in March of 2020, I believe. And if I had given it then, I think I had a slightly different perspective, because the pandemic has really brought to the forefront opportunities for using real-world data to learn about treatment effectiveness, vaccine safety issues in a way that I hadn't really thought about it prior to the pandemic. So I've been doing research on how to use EHR data to conduct research for about the past decade, but the pandemic has really prioritized these issues, and it's also led me to think more about the urgency with which we address clinical medical questions and whether or not and when real-world data can address these urgent questions. So as I just said, the pandemic has really elevated the prominence of using real-world data to answer clinical medical questions. And early in the pandemic, in particular, when clinical trials had not yet had a time to enroll subjects, collect data, conduct analyses, people started to churn out papers started to churn out results from real world data while we were waiting to learn from the clinical trials, which tend to be much slower. In short order, we saw a lot of publications about COVID-19 using real-world data, and then almost as rapidly a backlash against that real-world data-based research, kind of decrying its lack of rigor, its bias, and potential erroneous evidence that it was generating. So on the next couple slides, I have a few titles of editorials and perspective pieces that came out in 2020 and 2021, raising questions about how we were using real-world data to try to learn about medical product effectiveness. So this first one, the pandemic is prompting widespread use and misuse of real-world data. I certainly agree with that. We saw some good opportunities to use real-world data. We also saw some very questionable uses. There were also a number of commentaries that started to draw this dichotomy between the type of evidence and the quality of evidence that we generate with randomized clinical trials and real world data. So this one, can real world data really replace randomized clinical trials? I think the answer to that is no, as I will discuss throughout this talk. And then finally, the magic of randomization versus the myth of real-world evidence from the New England Journal of Medicine. So this one in particular is very concerning to me because it creates this sense that a randomized clinical trial is in some way magic. It's going to produce evidence that is in an entirely different category of quality and validity versus these real-world data studies, which are just, a myth. They're leading us to erroneous conclusions and not taking us in the right direction. So what I'm going to talk about today is how I think about generating valid evidence from real-world data, and also how we bring real-world data studies together with randomized clinical trials in order to address questions in ways that neither of these sources alone is particularly good at. So I want to think about real-world evidence potentially allowing us to expand the generalizability and external validity of clinical trials, and clinical trials as potentially allowing us to answer questions overcoming some of the data quality issues I'm going to discuss and confounding issues that arise in real-world data studies. So the final headline that I have here, randomized controlled trials versus real-world evidence, neither magic nor myth. That's the perspective I'm going to present you with today. I think each of these two sources has strengths and weaknesses, and we need to learn what those are so that we can bring them together in ways that are complementary. Neither is magic, but both have real potential to help us learn. So when I think about randomized controlled trials and real-world data-based studies, both have strengths and weaknesses. Clinical trials have historically been considered to be the gold standard for estimating treatment efficacy, and they continue to be held up as a standard that we are emulating using any other type of study. Nonetheless, trials are not without limitations, most notably limited external validity, especially when some patient populations are underrepresented. This is particularly true in the oncology context, where for decades, critics have been noting the fact that certain populations are significantly underrepresented in oncology trials, phase 3 trials, most concerningly. Particularly racial and ethnic minorities continue to be substantially underrepresented in phase 3 trials, and poor prognosis patients are also substantially underrepresented and in some cases explicitly excluded from oncology trials. Despite the fact that the trial may not necessarily include a representative patient population, once a treatment is approved, it tends to be prescribed broadly. So as I'll discuss later in my talk, I've done research with colleagues in oncology where we look at how newly approved treatments diffuse into care and who they're being prescribed for. And what we see is that even when, for instance, poor prognosis patients are underrepresented or excluded from trials, once new treatments are approved, they tend to be prescribed for those populations. So this is a place where we can potentially use EHR data to start to bolster the evidence that we get from trials alone. We can help to fill in this efficacy effectiveness gap and understand how treatments perform in real world populations receiving care as it's actually delivered in routine clinical practice, rather than relying on the somewhat more artificial and low generalizability setting of a clinical trial. However, as I've talked about for many years and tried to convince colleagues of, we also need to think really carefully and have significant caution when using real world data, because these data were not collected for research purposes and hence may not be research quality and may not be appropriate for answering all the different kinds of clinical questions that we might have. So most of my research focuses on sources of bias and how to overcome it that arise due to data quality issues in EHR and other real-world data sources. So in this slide, I've divided the types of bias that we might encounter in real-world database studies into information bias and confounding. But most of my research centers on the information bias side of the slide. And in fact, when I think about confounding, most of the time I'm thinking about issues that arise due to confounding, due to incomplete capture of confounding information or measurement error, misclassification, or other information bias issues in confounders. So the sources of this information bias in real world data sources, the reasons it's so pernicious and so ubiquitous arise because of the fact that EHR data are generated in response to the administrative, clinical care delivery, and billing needs of the healthcare system, not our research needs. So in research, if we're designing an observational study, we typically would specify all of the data elements that we need in order to conduct our study, and we would have precise and specific definitions for each of those data elements. And that's very much not what we have when we extract data from the EHR. So measurement error and misclassification are going to be commonplace because of lack of harmonization in how individual providers and individual healthcare systems are using medical codes and other terminology. There's a lack of common definitions. We're often relying on structured data elements, so ICD codes or procedure codes that may be coarse or non-specific. They might not have the degree of granularity that we really need to conduct our research. And additionally, missing data is ubiquitous and quite complex in structure. On the other side, when we think about confounding, there's going to be a lot of missing confounder information. Many of the confounders that we would really like to be able to account for in our analyses are simply not captured in a consistent or structured way in the EHR. So we have limited information on behavioral risk factors and social determinants of health. Sometimes these pieces of information are captured in narrative text notes. And I think the research area of extracting information on social determinants of health from narrative text is a very active and very interesting area. But in addition to the fact that it's hard to do, that information is just not systematically captured. So even when it is there, it raises the possibility of informative missingness. Why were these data collected for this individual and not for somebody else? There's all sorts of other information like symptoms and family history that is sometimes available, but is not consistently collected. And even when this information is available in the EHR, we still have the same problem as illustrated on the left-hand side of this slide, that we don't have consistent usage of definitions, common usage of definitions. So there are going to be information bias issues that persist due to measurement error and misclassification in confounder variables. So taking all of those things into consideration and thinking about the fact that trials have strengths and weaknesses, real world data has strengths and weaknesses, what I'm going to talk about today is how we can bring these two things together to try to amass the strongest evidence base possible for helping us to learn in a timely fashion about clinical medical questions. So I'm going to discuss three specific areas. First, I'm going to talk about data provenance and EHR phenotyping. I'll then discuss how we can take phenotypes extracted from the EHR and come up with estimates that are minimally biased given the imperfections in those phenotypes. And then finally, I'm going to give an example from my research where we thought about how we could bring together evidence from a trial with evidence from real world data to try to answer questions in a way that neither data source alone could really do. In this slide, I have a depiction that comes from Google Labs of the EHR data provenance process as illustrated for a single individual. So here we have a representation of the process of one person interacting with a healthcare system. You see that this person over time is making encounters of different types. So they're making a visit to their primary care provider, they have an urgent care visit, a hospitalization. And at each of those encounters, different types of information are collected. So sometimes they have lab tests performed. Over time, different lab tests at irregular intervals. They have medication prescriptions that are written, their vital signs are assessed, various diagnoses are recorded. So we could think about taking this pretty complicated and heterogeneous process of the patient interacting with the healthcare system and the data footprint that it generates and formatting this into a data set. And what this data set is going to look like is a longitudinal observational data set with repeated measures across an individual and with a lot of missingness. So at any given visit, we're going to have some data elements that were recorded and other data elements that weren't recorded. This is one person. Every person in our healthcare system database is generating a similar digital footprint. We could take all of their data, make it into one big longitudinal observational database. And if we just look at it superficially, if it just showed up on my network drive and I log in and I go to analyze the data and I don't know of anything about where it came from, it would look a lot like the data from any other longitudinal observational study. But it has many important differences because nothing about this process of the patient interacting with the healthcare system was pre-specified by me, the researcher. So the timing of the patient's interactions, the pieces of information that are collected at the time of each of those encounters are entirely separate from my research needs. The frequency of these visits might be inadequate to capture the types of transitions between disease states that I'm trying to capture. They might not have the tests performed at the timing that I need those to be assessed, and so on. And every single patient contributing to this database is going to have their own data generating process that's attributable to their own pattern of interaction with the healthcare system. This is a very messy database with a complex missing data structure. And this process of interaction with the healthcare system violates many of the standard assumptions of many of the statistical methods that we use for longitudinal data and time to event data. Specifically, the process of interacting with the healthcare system is driven by the health needs of the individual. And many of our standard methods rely on an assumption of independence between the observation process and the underlying disease process. That's very much not the case in EHR data, where the reason that a patient is making a visit to the healthcare system is directly because of their individual health needs. So given this messy, heterogeneous, longitudinal observational database, what I, as a researcher, would like to do is extract data elements from that database so that I can start to answer the research questions that I'm really interested in. And that process of extracting data elements from the EHR, I'm going to refer to broadly as phenotyping. So phenotyping generally describes the process of characterizing the patients that are in our data sets using the available data. There's an enormous literature in informatics and to a lesser extent in statistics on EHR-based phenotyping. And the reason that there's so much written about this is that there are really no gold standards for any patient characteristic in the EHR. Almost everything, if you think hard enough about it, is error prone or a silver standard or not quite exactly the thing that we really wish it was when we derive it from the EHR. So almost anything that we want to characterize about our patients, but particularly their disease outcomes, are not collected in a gold standard way in the EHR. So we need to come up with some kind of way to deduce those characteristics since they're not explicitly recorded. This is tricky because of these data provenance issues, because each patient has their own unique frequency of visits and specific data elements that were collected. And we need to figure out how we're going to deal with that in order to come up with a phenotype that performs well for all patients. This is an example of a pretty traditional rule-based phenotype for type 2 diabetes that was proposed by a group called the eMERGE Consortium back in 2012. And in their data sets, at least when this was validated within eMERGE, they found that this was a very high-performing, high-accuracy phenotype. So basically what this phenotype is doing is querying an individual's EHR, and within that individual patient's EHR data, it's looking for diagnosis codes. So back in 2012, they were looking for ICD-9 diagnosis codes for type 1 and type 2 diabetes. It's looking for prescription medications related to the phenotype of interest. So looking for insulin, looking for type 2 diabetes medications. And it's also looking for abnormal biomarker values. So it's going to take the information that we find there related to diagnosis codes, prescriptions, and biomarkers, and it's going to use that to classify the patient as to whether or not they have the phenotype of interest, type 2 diabetes. This is a pretty standard way to construct a phenotype. This is pre-specified by the researchers based on knowledge of the clinical condition, how it's treated, and how it's coded. But it's deficient in a few ways that we would like to improve upon. So first of all, this phenotype doesn't take into account in any way the depth of information available for a patient. So thinking back to the figure that I showed a few slides ago, each patient has a very different depth of information. The one that was in that figure had a lot of interaction with our healthcare system. We might be applying this phenotype to someone like that, but lots of encounters, who had their biomarkers measured. We might be applying this phenotype to someone who made only a small number of visits to the healthcare system and about whom we really have very limited data. But if we just apply a rule-based phenotype like this, at the end of the day, we just come up with a binary classification that says you have type 2 diabetes, yes or no, and doesn't take into account the fact that we can make that classification with much greater certainty for some patients versus others. The other limitation of this type of phenotyping approach is that it treats normal biomarker values the same as absence of information on a biomarker value. So when we get down to this node in the phenotype, abnormal glucose or hemoglobin A1C, if a patient doesn't have any assessment of their glucose or hemoglobin A1C, we would say no, they did not have an abnormal glucose. Similarly, if the patient did have their glucose or hemoglobin A1C measured and it was in the normal range, we would say no, they did not have an abnormal glucose or hemoglobin A1C. So those are actually two very different things. The patient for whom we know that their biomarkers were normal, we have a lot more information than the patient who was simply never assessed. And that, in combination with the fact that whether or not the biomarker was assessed is probably related to signs and symptoms of the disease, which themselves are potentially informative of whether or not the patient has the condition, really should be taken into account in our phenotyping. So on this slide, I'm illustrating what is potentially a missing not at random missingness mechanism for the availability of biomarkers in the EHR data using the specific case, the specific example of type 2 diabetes. So typically, when we think about biomarkers or any other piece of data that's going to be inconsistently collected for patients in the EHR, which is almost everything, we would like for the assessment of that piece of information to be related to characteristics that we observe. In other words, we would like it to be missing at random. And if biomarkers were assessed based on risk factors that are observable to us based on EHR data, such as biometric measurements that are readily available like BMI, or risk factors like age and race, which are quite frequently collected, then that would represent a missing at random missingness mechanism. However, what's more often the case, given the complexities of EHR data provenance, is that availability of more detailed information like biomarker measurements is related to the healthcare seeking behavior of the patient. So all sorts of factors affecting access and utilization, like whether or not a patient has health insurance, what their health literacy is, whether or not they know the symptoms of the specific condition. All of those things are going to be related to whether or not they choose to make an encounter, whether they discuss the condition with their provider, and therefore are receive an order to have their biomarkers measured. So healthcare seeking behavior is going to be related to availability of biomarkers. And additionally, healthcare seeking behavior is going to be related to the underlying condition itself through connections between socioeconomic status and many conditions that we're interested in, such as type 2 diabetes, and so on. So as a result, we end up with a relationship between these unobserved healthcare seeking behavior characteristics and missingness in biomarkers. There's also going to be a direct relationship for the unobserved true patient phenotype and the biomarkers through things like signs and symptoms that the provider knows about, the patient knows about, and that lead therefore to whether or not the patient has biomarkers assessed, but which aren't explicitly captured in the EHR. So in many, many cases, if you think hard enough about the data provenance process, you'll be able to convince yourself that there's at least a pretty good chance that there is a missing not at random missingness mechanism for many of the data elements that we extract from the EHR. To try to deal with some of these challenges, a few years ago, my group proposed a latent phenotyping model. The idea behind this being that we wanted to explicitly acknowledge the fact that these phenotypes that we're trying to characterize are never explicitly observed in the EHR. So we proposed that the phenotype should be conceptualized as an underlying, unobserved, or latent characteristic. So in my notation here, the phenotype is represented by Y. We assume that Y is not observed for anybody. That's your true phenotype. All we know is what turns up in the EHR, which is at best a poor representation of the truth. So Y is your true phenotype. It arises from a Bernoulli distribution with parameter theta I, which is your subject-specific probability of having the phenotype of interest. We assume that the phenotype is completely unobserved for everybody, but what we do observe are a bunch of features, a bunch of signs and indicators in the electronic health record, which give us clues as to whether or not you have the phenotype of interest. So we denote these features using X, and here I'm using D to just denote a generic distribution, whatever is appropriate for the specific type of feature under consideration. So X is going to be a feature that comes from a distribution with parameters that differ depending on which underlying true phenotype state you're in. So for instance, X might represent a biomarker that comes from a normal distribution where the mean of that normal distribution is different if you have the phenotype compared to if you don't have the phenotype. An important thing to acknowledge in the EHR is that not only are these observed characteristics informative about the underlying phenotype, but presence or absence of some of the inconsistently collected features is also informative. So in the example of type 2 diabetes, we have biomarkers, glucose and hemoglobin A1C, that are informative about type 2 diabetes status, but also whether or not those biomarkers were measured is informative. And it's reasonable to assume that individuals with diabetes will have their biomarkers measured more frequently. They'll be more likely to have information about their biomarkers than individuals without type 2 diabetes. So missingness in the biomarkers is also assumed to be a feature that we can incorporate into our phenotyping. We propose this model in a Bayesian estimation framework. And the really nice thing about the Bayesian framework for this particular problem is it allows us to assign priors for our model parameters. For instance, these mu's that encode existing information from the clinical medical knowledge base about how these features are expected to differ for individuals with and without the phenotype of interest. For instance, in the case of glucose, we know the operating characteristics of glucose with respect to diabetes. We know how mean glucose differs for individuals with and without diabetes. So we can encode that information via a prior distribution. When we take all of these features together, they give us a likelihood that's a mixture likelihood. So we're mixing over the two phenotype classes. We put that together with our priors, and that's going to allow us to get the posterior distribution for the individual specific probabilities of having the phenotype of interest. So at the end of the day, what we're going to come up with is a posterior mean for theta I, which will be the individual's probability of having the phenotype of interest. We applied this in the context of an EHR-based study using the PEDSNet data, which is one of the PICORNet networks. We looked at children who were aged 10 to 18 years of age with 2 clinical encounters. So we wanted to have some minimum depth of information from the EHR. And because type 2 diabetes is quite rare in children, we wanted to focus on a cohort who were at high risk for the condition. So we required that they had to have a BMI Z-score in excess of the 95th percentile for their age and sex. I'm going to show you data from 2 of the sites in the PedsNet network, 30,000 kids from one site, 24,000 kids from the other site. This table is illustrating some of the features of these kids that relate to type 2 diabetes from the 2 sites. So these are the features that went into our phenotyping algorithm. And the thing that I think is really interesting about this information is that it illustrates both likely differences in the prevalence of type 2 diabetes between the 2 sites, as well as differences in practice patterns and coding patterns between the 2 sites. So when we look at the EHR and we think about ways that we can use these data to start to learn about type 2 diabetes, there are all kinds of different pieces of information that we might pull out. We could pull out the biomarkers, and we see there's a difference in mean biomarkers between the sites, suggesting maybe a higher prevalence of diabetes at site B. We could pull out utilization information. So how many of these kids made a visit to an endocrinologist? And we see huge differences between the 2 sites in terms of those utilization patterns. We could also take a look at prescription medication use. Diagnosis codes is an obvious place to go in terms of phenotyping a condition of interest. And as I mentioned, we can look at missingness patterns. So how frequently are the biomarker tests that are going to be most informative about diabetes actually being performed in these kids? And we see across all of these things, big differences between the 2 sites. So we know that there are differences in prevalence, but we know that there are also different practice patterns and different coding patterns going on here. And then finally, if we take the emerge algorithm and we apply that rule-based phenotype in this setting, we come up with estimates of 0.3% and 0.9% for the prevalence of type 2 diabetes in these cohorts. And that's almost certainly an underestimate in these high-risk kids, probably due to the fact that that algorithm was not designed for children. It was designed for adults, and consequently is probably not incorporating information about type 1 diabetes in a way that's appropriate for this population. So really motivating the need to tailor the approach that we're using to the specific clinical context and to the specific sites that we're looking at. We applied our latent phenotyping approach. We come up with estimates for all of the model parameters describing how our features differ between kids with and without diabetes. And that allows us to characterize things like, what was the mean shift in the biomarker for children with or without the underlying true phenotype? What's the sensitivity and specificity of each of the different binary features that we've incorporated into our phenotype algorithm? And so on. And the ones that I've illustrated here in red are those where the 2 sites differ quite a bit. So for instance, the specificity of making a visit to an endocrinologist is 94% at site A, but only 84% at site B. And the reason that I've highlighted that here is really to kind of lead us into the next part of my talk, where I'm going to discuss issues related to differential measurement error, differential misclassification in EHR-derived elements, and really thinking about the fact that due to differences in the available data across patients and across providers and healthcare systems, it's really necessary to limit assumptions about transportability of phenotypes across sites, because we do see just really different performance of different phenotypes in different settings and different patient populations. This particular approach to phenotyping, as I said, gives you a posterior probability of having the phenotype of interest for each child. And here's what those distributions look like for site A and site B. What we're hoping to see here, because we have 2 classes, is good separation of the phenotype classes. So we'd like to see one group with low probabilities and another group with high probabilities of having the condition of interest. We're seeing some separation here for site A, a little bit poorer separation for site B, and overall a general shift in the distribution for site B relative to site A, representing that slightly higher prevalence. So we can take these posterior probabilities and propagate them forward into subsequent analyses. So we can use these probabilities as phenotypes in subsequent analyses that allow us to characterize the patients, but also capture our certainty about our phenotyping. So a child for whom we're assigning a phenotype of probability of diabetes of 0.001% is a child that we're very certain does not have diabetes, whereas a child that we're assigning a phenotype probability of 0.01 or 0.02, we're really not sure. And we can move those probabilities forward into subsequent analyses to capture that uncertainty about the phenotype. So to wrap up this first part of my talk, the main thing that I'm hoping to have conveyed to you is that EHR contains very heterogeneous information across individuals and across healthcare systems. And in order to use that information in a way that's going to lead to valid evidence, we have to make sure that we have adequate systematic data capture for all of the data elements that we need in order to conduct our analysis. Phenotyping accuracy, which essentially means the accuracy of every data element that's going to be part of our subsequent analysis, is dependent on the data generating mechanism or the provenance that gives rise to the data that we see in the EHR. And there's a high risk of differential misclassification and missing not at random missingness when we're using data that arise from routine clinical medical interactions that we really should be accounting for in the phenotypes that we propose and the subsequent analyses that we conduct. And then finally, taking me into the next part of my talk, transportability of phenotypes may be limited because there is so much heterogeneity and because there are so many differences between different healthcare systems in terms of what they record, when, how frequently they see patients, what their coding practices are, and so on. So that was one example of how we can extract phenotypes from the EHR. As I said, there's huge literature on this. There are many, many ways that we can extract phenotypes from EHR-based studies. But at the end of the day, we're going to end up, no matter what we do, with a phenotype that we know does not agree perfectly with the underlying truth. And what we would like to do, our objective, is to obtain unbiased estimates in analyses that are using these imperfect EHR-derived phenotypes. There are a number of things about the EHR setting that make that a particularly challenging task. For one thing, validation data are costly to obtain. So in many settings with measurement error and outcome misclassification, what we need to do is sample the data and obtain a gold standard for a subset of individuals. But in the context of real-world data or EHR data, what validation data consists of is conducting manual chart review to obtain the best possible information on the characteristics of interest. Manual chart review is slow, manual chart review is expensive, and if we're working with data sources like big multi-payer claims databases, it may be impossible. We may have no ability to go back to the original medical record data. So validation data are precious and sparse in the EHR setting. As I mentioned a moment ago, operating characteristics of our phenotypes may vary widely between the data set in which the phenotype was developed and other data sets. So we don't want to make assumptions that our phenotype has known sensitivity and specificity, or that sensitivity and specificity is necessarily independent of patient characteristics or healthcare system. And then finally, as I've mentioned, because data provenance for EHR data is so heterogeneous, there's a high risk of differential misclassification. So we want to make sure that we're using methods that account for the possibility of differential misclassification. The specific inferential task that I have in mind is that we have some outcome of interest. And here I'm going to use an example from a study of second breast cancers that I participated in a few years ago. So our outcome was whether or not a woman with a personal history of breast cancer experienced a second breast cancer event, or SBCE. We wanted to identify risk factors for SBCE, like age at diagnosis, stage of the primary breast cancer, hormone receptor status. So what we would like to do, thinking of some kind of generalized linear model type of estimation framework, we'd like to estimate these parameters beta that describe the relationship between a second breast cancer and patient characteristics. However, working with real world data, we don't observe second breast cancer status for any of our patients. What we observe is a second breast cancer phenotype that we have developed and validated and we believe has good operating characteristics, but does not necessarily agree well with the truth. So our SBCE phenotype is related to the underlying truth, of SBCE via some sensitivity and specificity. And here, sensitivity and specificity, I have indexed by X to reflect the fact that the performance of our EHR-based phenotype may vary according to patient characteristics. If we look at the relationship between S and X, we're going to be estimating beta star, something that is not quite beta. It'll tell us about the relationship between patient characteristics and the EHR-based phenotype, but due to the imperfections in that EHR-based phenotype, we can't assume that beta star is equal to beta. There are a number of different ways that we can think about this problem. And if anything, what I hope to leave you with from this part of my talk is the importance of thinking about this problem, doing something, whether it's an analytic approach that addresses outcome misclassification or quantitative bias analysis, but thinking in some kind of explicit way about the fact that our phenotypes are imperfect rather than just sweeping it under the rug. So when we think about the data that we have available for analysis, if we've conducted a validation study, we went back to the charts, we obtained gold standard labels for a subset of our individuals. We could think about using those data to obtain estimates of beta. So what I'm going to call model 1 here is the very, very conservative approach of saying, because we don't have access to gold standard outcome information from the EHR, we're only going to analyze the individuals in the validation subset. We'll take their gold standard labels Y and regress them on patient characteristics X to obtain consistent parameter estimates for beta. And here the little subscript V is representing the fact that I'm doing this only in the validation subset. So this is going to give me a consistent estimate for beta, but a very imprecise estimate because I'm relying on just this small manually validated subsample. A person who is much more enthusiastic about real world data, who feels great confidence in their big EHR sample and doesn't want to throw all that information out and just rely on the validation data, would take approach 2. So model 2 here says, let's use the full sample, here denoted with the subscript F. We'll just take the EHR-based phenotype S that we're able to generate for everybody and regress that on X. And there's more data here. It's big data is going to solve all my problems. So I'm just going to regress S on X and I'll get estimates of beta star, and I feel good about that. So that's an approach that's going to lead to a very precise estimate, but beta star is not consistent for beta. So we're going to get good precision, but at the cost of a parameter estimate that is not targeting the correct parameter. Model 3 is a classic approach to outcome misclassification. So way back in the 90s, Magder and Hughes proposed a maximum likelihood-based outcome misclassification approach. It's very straightforward to implement. From my perspective, it's quite substantially underutilized in EHR-based studies. This is an easy way to account for outcome misclassification when we are willing to make an assumption about what the misclassification model looks like. So in the results that I'm going to show you, the specific assumption that I'm going to make is that there's non-differential outcome misclassification. So I'm going to assume there's a single sensitivity and specificity that holds for my entire sample. You could come up with a more complex outcome misclassification model than that if there are specific factors that you believe might influence the sensitivity and specificity of your phenotype. But the bottom line with model 3 is we have to have correctly specified that misclassification model. The approach I'm going to contrast this with, I'm going to call model 4, was developed by a student working with myself and Yang Chen at the University of Pennsylvania, Jesse Tang. And Jesse Tang's approach, which here is called augmented estimation, is a way to combine beta hat V, our consistent but not very precise estimate based on validation data, and beta hat star F, our very precise but not consistent estimate based on the surrogate in the full sample. The really clever thing about this approach that Jesse developed is that it accommodates differential or non-differential misclassification without requiring explicit specification of a misclassification model. So given the complexity of EHR data provenance, we really don't want to have to be able to say, here are all the factors that affect the accuracy of this phenotype. I know what they are. I can write down a model for that. The way that we're going to do that is by defining one additional regression, which is regressing the surrogate, the EHR-based phenotype S, on our patient characteristics in the validation subsample only. By doing that, we can then construct what's called here model 4, or the augmented estimator, which is a weighted combination of beta hat V and beta hat star F, where the strength of the weights, how much faith we put in each of those two estimates, depends on the correlation between beta hat V and beta hat star V. So in the validation sample, if our estimate based on the surrogate looks a lot like our estimate based on the true gold standard outcome, then that means that signals to us that our surrogate is quite good. And in our weighted combination here, we're going to end up putting all of our weight on beta hat star F. Conversely, if beta hat V and beta hat star V don't look much like each other, that means our surrogate is probably not very good. It doesn't agree well with our gold standard label Y. And in that case, we want to rely primarily on beta hat V, our estimate in the validation sample only. These are the results of a simulation study that we conducted using the data from the study of second breast cancer events, in which we had gold standard labels for everyone in the study cohort. So we had gone back to the charts, done manual chart review, obtained a gold standard for everyone. And so what we did was to sample repeatedly, construct validation subsets of varying sizes, and then apply each of the methods that I just described, pretending that we only had validation data for those subsamples, the sample sizes of which are given on the x-axis here, and compare the estimates that we got to the estimate that we would have gotten using the gold standard for everybody. So the vertical dotted line is the estimate based on the gold standard in the full sample, and the gray band is the 95% confidence interval for that estimate. In the interest of time, I will try to summarize these results relatively quickly. The basic idea here is that model 1, which uses only the validation data, performs the same regardless of the quality of our surrogate. So the 3 panels represent 3 surrogates with different operating characteristics and with non-differential or differential outcome misclassification. Model 1 doesn't use the surrogate, so it always performs the same. It doesn't matter how good our surrogate is. But if our validation sample is small, it's going to be very imprecise and it's going to be pretty unstable. So we might end up with an estimate that's fairly far away from what we would have gotten if we had the gold standard for everyone. Model 2, which relies exclusively on the surrogate, is going to give us a very precise estimate, but how close that estimate comes to our gold standard estimate depends on how good our surrogate was. So in the upper left, we were using an example of a surrogate that has very good operating characteristics. Model 2 performs great. When we move to the upper right and lower left panels, there we have surrogates that are not as good, and we see that estimate start to deviate from what we would get with the gold standard. So essentially, we're placing all of our faith in model 2 on the quality of our surrogate. Model 3, which uses an outcome misclassification correction that assumes we know the misclassification model, performs well when we correctly specify the misclassification model. So in the upper 2 panels, we have non-differential misclassification. That's what model 3 is assuming here. And so it performs quite well. In the lower panel, we used a surrogate that exhibits differential misclassification, and model 3 there performs quite poorly. And in fact, when we misspecify the outcome misclassification model, making the outcome misclassification actually performs more poorly than if we had made no correction at all. And then finally, the augmented estimation approach is basically a trade-off. It's making a trade-off between model 1 and model 2. So it gives us estimates that are more precise than model 1, but also generally closer to the estimate based on the gold standard than we would get with model 2, regardless of the structure of the misclassification. So model 4, because it is data adaptive and is responding to the observed misclassification pattern in the actual data set, tends to perform well across different surrogates. The one caveat being in the lower left here, we see in this validation subsample that's small, where we have a differential surrogate, the point estimate actually deviates from the gold standard quite a bit. We observed this across a number of permutations of these simulations. And it would seem that what happens in that setting is we have a small validation sample and a complex outcome misclassification process. And in that setting, the model isn't able to estimate that covariance matrix well, and as a result ends up with a poor estimate. So I like to be forthright about the strengths and weaknesses of methods. Nothing is a panacea. And in this case, we found the augmented estimation approach works quite well across many settings, but is limited in the setting of small validation sample. To wrap up the second part of my talk, I will just reiterate that sources of measurement error and misclassification are ubiquitous in real world data. They're everywhere. We have to acknowledge that they exist. We have to account for them in the statistical methods that we're employing and not try to gloss over them and just write the one sentence in the discussion section that I see in so many papers that say, yes, there might have been outcome misclassification, but if so, it probably just resulted in bias towards the null, so we're not really going to worry about it. That's really not adequate in the context of real-world database studies where measurement error and misclassification are complicated. They derive from the complex data provenance process, and so assuming that we're looking at non-differential outcome misclassification is very naive at best, I would say, and we really need to be explicitly accounting for it in our analytic methods. So finally, to return to where I started and thinking about how we can take real world data, which does have all of these imperfections and challenges, and use it to help us learn, to help us generate evidence and reach conclusions that we wouldn't be able to reach using randomized controlled trials alone. Just wanted to give an example from research that I've been doing with collaborators in oncology that's got me thinking along these lines of how do we bring these two data sources together. The specific context that I was working in was evaluating the comparative effectiveness of treatments for advanced urothelial cancer. So this is a setting where prognosis is quite poor. 1 year survival is about 40%. At the time we were conducting a study, immune checkpoint inhibitors, or ICIs, also called immunotherapy, had recently come on the market as a treatment that offered potential for survival benefit. But the evidence about effectiveness and particularly comparative effectiveness of ICIs was limited because these treatments had been approved by the FDA based on single-arm phase 2 trials. So there was a suggestion that ICIs offered benefit, but they had not been compared head to head against the standard of care at the time, which was chemotherapy. When I discussed this study with my oncology colleagues, at that time, phase 3 trials were underway. They were going to give us the evidence that the clinicians wanted on the comparative effectiveness of chemotherapy and ICIs, but they were taking a long time. They were slow, and these drugs were already on the market, giving us the potential to conduct a real-world data-based comparative effectiveness study comparing chemotherapy and ICIs. And in that way, we could use real-world data to try to inform decision-making in the interim while we waited for the trials to complete. So we could try to do what people have been doing during the pandemic for COVID-19 and kind of advance the evidence base a little bit more rapidly while we wait for clinical trials to catch up with what's happening in the real world. So using data from the Flatiron Health Oncology EHR database, we conducted this study of the comparative effectiveness of ICIs and chemotherapy for treatment of advanced urothelial cancer. And what we found was that early in follow-up, immunotherapy had a hazard ratio greater than 1, poorer survival outcomes than chemotherapy. Later in follow-up, immunotherapy had hazard ratio less than 1, overall survival that was superior to chemotherapy. We published those results in European Urology in 2019. On the left here, I have a figure from that paper illustrating the Kaplan-Meier curves for overall survival for chemotherapy and for immunotherapy. You can see the poorer performance of immunotherapy early, and then this potentially durable benefit in a subset of patients that leads to good outcomes later. On the right-hand side of this slide, I have results from a paper by Galsky and colleagues that was published about 6 months after our 2019 paper, with the results of the phase 3 randomized control trial comparing these two treatments head-to-head. And you can see they came to a rather similar conclusion in terms of the relative performance of these two treatments. So what we had accomplished was essentially moving the timeline forward, advancing the evidence base by about 6 months in terms of allowing clinicians to make informed decisions based on head-to-head comparison of these treatments while we were waiting for the clinical trial to conclude. And ultimately, we came to very similar conclusions to what the trial found, but we can feel good confidence in the results that we had generated earlier because of the fact that once we employed randomization to address some of the confounding issues and to address and used a randomized control trial to address some of the data quality issues, we still came to a very similar conclusion. This got us thinking about whether or not we could develop an approach to explicitly compare results between RCTs and real-world database studies to allow us to draw conclusions about how outcomes differ between the real world and between the RCT setting, and to also think about how results differ in those different populations, the population of patients who are participating in trials and the population of patients who are being treated in the real world. Because as I said early in this talk, those two populations are not necessarily the same. So working with another student at Penn, Kylie Getz, we developed a two-step pipeline to carry out explicit comparisons of real-world data results and randomized controlled trial results. The idea behind this two-step pipeline is that as academic researchers outside of the pharmaceutical companies who hold the individual level data on the randomized controlled trial results, what we have access to is individual patient data from the EHR and aggregate data from the trials in the form of outcomes published using Kaplan-Meier curves and marginal patient characteristics, so distributions of patient characteristics in the forms of means and proportions, such as we would see in a table 1. So what we would like to do is take that aggregate trial information, combine it with the individual patient data from our EHR, and see if there are differences in outcomes or even in comparative effectiveness of treatments between what we see in the real world setting and the trial setting. As I said, the pipeline consists of 2 steps. The first step is a survival data reconstruction step. So what we're going to do is take the published Kaplan-Meier curve, use curve digitization techniques in combination with an approach proposed by Guillot and colleagues back in 2012 to generate individual level survival data from a published Kaplan-Meier curve in order to create individual level survival data for the trial. Once we have individual level data on the outcome from the trial, we can put that together with our individual level outcome data from the EHR, but we know that there are differences in patient characteristics between those two populations. So what we would like to do is standardize our EHR population so that their distribution of patient characteristics looks just like the distribution reported in table 1 for the trial. And I think quite interestingly, we can either choose to completely standardize our EHR population so they look exactly like the trial, or we could choose to standardize them with respect to all except a subset of differences. For instance, we might leave residual differences in patient demographics, and then we could take a look at differences between the trial and the EHR and attribute remaining differences in the outcome to those characteristics that had remained unstandardized. So we propose to do this using the method of matching adjusted indirect comparison weights, which are basically just weights that are going to balance the distribution of marginal moments between our real world data sample and our trial sample. In the interest of time, I'm going to summarize this pretty quickly, but we, as a trial, I should use a word different from trial, as a exemplar, we applied this approach to a study of a chemotherapeutic treatment for urothelial cancer, where we had data on the trial from a published paper, and we brought that together with data for a similar patient population from the Flatiron Health Oncology EHR database. When we look at the distribution of patient characteristics for the trial patients and the EHR patients, they're pretty similar, with the one notable difference that performance status was somewhat better, somewhat surprisingly, in the EHR sample compared to the trial sample. So what we're going to do is construct weights that balance these two distributions, so our EHR patients look just like our trial patients. When we do that and then compare their outcomes, here I have the Kaplan-Meier curve for the trial and a weighted Kaplan-Meier curve for our EHR-based sample, as well as results of a weighted Cox proportional hazards model comparing outcomes in the two. And you can see visually they look quite similar. The hazard ratio that we obtain is also quite close to 1. So here, when we compare two patient populations that are very, very similar, receiving the same treatment, standardized so their patient characteristics are the same, outcomes in these two populations look the same. And this creates a basis for us moving forward to conduct similar studies in which we look at relative effectiveness of two different treatments in a trial compared to EHR, or standardize our EHR population to all but a subset of characteristics relative to the trial population, so we can start to pinpoint which differences in patient characteristics lead to the biggest differences in outcomes between trials and EHRs. All right, I will wrap up there. I see I'm a little bit over time. The thing I hope I have left you with is that real-world data can help us to generate evidence in a timely fashion, responding to the urgent need for new knowledge. But in order to do that, we really need to think carefully about data quality issues, measurement error, misclassification, all the stuff that I've talked about, because it's very easy to use real-world data to come to incorrect answers due to challenges and limitations of the data. And I think doing this in an interdisciplinary way, where we're working together as data scientists, partnering with clinicians, is really key, because we do not have the depth of knowledge that they do about what the visit process looks like, about when tests are being performed and why, and so on. We can conduct good studies with real-world data. We can conduct bad studies with real-world data. And the role that I really see for statisticians is holding us to the highest possible standard for the quality of evidence that we're going to generate, whether it's from real-world data or from randomized controlled trials. And we can do that through contributing our understanding of study design, how that fits in with data provenance, how that fits in with the analytic methods that we're using to make sure that we are advancing the evidence base in the highest quality way possible. We'll end just by acknowledging, in particular, these are all students and postdocs, some of whom are now faculty members elsewhere, but were students and postdocs when they worked with me at University of Pennsylvania, and none of this research would be possible without them. And thank you for your time and for your attention, and my apologies for running long.
No problem. That was fascinating. And we thank you so much for, I think I personally learned a lot in this, and it was a really a great overview of the different approaches of where you can use these methods. I think since we are over, I'll hold it open for like a few, 20 seconds to see whether anyone has some questions. Because I think people are going to other meetings, so we do seem to be losing people, right?
It's a back-to-back Zoom environment. I'm very responsive on email, so I'm sorry that I didn't leave time for questions, but if there are any questions, I'm always happy to answer email. You can feel free to write to me.
Okay, thanks. We really appreciate that. Thanks so much, Dr. Hubbard. It was great having you here, and hopefully we'll be able to have you again in person.
That would be wonderful. I'm looking forward to it.
Thanks so much, everyone.